Sure. Okay. So the beginning will just be the information that you can see on this slide as well. So it's my pleasure to show you some of the, first of all, some of the organizational information for the second round of our emerging topics in biomolecular magnetic resonance. Today we again have two speakers. First will be Anne McDermott, followed by Hashim Al-Hashimi. And just a quick reminder that we aim for a Gordon Research Conference policy, so no private video recordings, please. And these lectures anyway will be recorded and put up onto accessible YouTube channel. Oops. So, and the website has also just gone live, so this is a much easier way to access all the information, the videos, upcoming speakers, and the schedule at emergingmr.org. And there you'll also be taken to the link. The videos are actually hosted on the YouTube channel of ICMRPS. Just a note from last time for everybody to use the Q&A rather than the chat for asking questions. This lets all of you also read other people's questions easily and upvote questions to the top that you find to be the most interesting, so that we'll read those out first. So in the interest of time, I'll just give a very quick introduction. It's my pleasure today to introduce Anne McDermott. She did her PhD in chemistry at UC Berkeley, where she worked with Kenneth Sauer and Melvin Klein, and subsequently did postdoc training at MIT with Robert Griffin. She's joining us today from Columbia University Department of Chemistry, where she's professor of biological chemistry, and has many interests in membrane proteins, which are close to my heart, also in DNP, which she did some nice work on a viral coat protein recently, and of course, method development in solid state NMR. And today, she's going to tell us a little bit about another topic, I guess, which I haven't heard about yet, which is the putative amyloids in human health. So I'm looking forward to your talk. And the stage is yours.
Lauren, thank you so much. You can hear me?
Yes.
I'm going to share my screen. Really, thank you for that overly kind introduction. I will talk about proteins with key roles in signaling, in stress response, in cell death, in immunity. These are multi-domain proteins that combine classic globular functional domains with low complexity domains. And for their ill-behaved nature, they have received a treatment of solid state NMR. And in that, we are trying to sidestep the divide and conquer strategy and study some full-length systems. And before I jump into the details of that, I'd like to there we go. I'd like to just take a moment to thank all of you, Marcus, Lauren, Stefan, Christian, May, Oscar, and Art. You've done a very nice thing for the community to set this up, and I'm humbled by being one of the speakers. So thank you ever so much. So the first protein that I'm going to talk about manages a cell death pathway that might not be that famous or that well known, called necroptosis. Let's start out talking about necrosis versus apoptosis, the famous cell death pathway. They're clearly distinct. And with necrosis, maybe it's in poor taste, but let's think about the US and our response to the COVID pandemic. Nobody's in charge. Okay, the cell is overwhelmed by the insult, the volume expands greatly, mitochondria become swollen, the plasma membrane ruptures, and everything is kicked out. And for that, we get a strong inflammatory response, perhaps an international inflammatory response. Apoptosis, on the other hand, the famous cell death pathway, is a very orderly affair, which we can think about with maybe some more well-organized governments and our friends in East Asia or in Europe. proteases and nucleases within the confines of an intact plasma membrane disassemble the cell, and it gradually shrinks, leaving no trace. A kind of hybrid of the two caught many people by surprise, and that is a programmed necroptotic pathway. So it has some properties in common with apoptosis in the players, in the fact that it's a response to insults such as UV, X-ray, temperature, chemical. We're going to think about high pathogen load here. That's an important example. And upstream, it might be elicited by the tumor necrosis factor being ligated, trimerizing at the membrane. But the issue here is that there are many downstream paths that are possible. And we'll talk about the receptor interacting protein kinase, in a cheeky way referred to as a RIP kinase for these cell death pathways. That's a master regulator, one of the few proteins that's truly an essential protein. And it's been a very, very important drug target, and it has a role in deciding and sort of regulating which path we're going to go down here. It's thought about in the context of neurodegeneration, autoimmune diseases, cancer, infection, and other contexts. So another point of contrast is that largely apoptosis is a story of proteases, caspases, a cascade of them. Necroptosis is a story of kinases. And it ends with a pseudokinase. MLKL has a kinase-like domain that's not active, and it becomes phosphorylated by another kinase, RIPK3, and that creates a situation where it oligomerizes and pokes holes in a membrane. And so that's a way in which it's like necrosis. It ends badly with holes in the membrane, but other than that, it's a very programmed affair. So I meant to say on the last slide that there is a commitment moment where RIPK1 and RIPK3 interact, and they form a complex, sometimes referred to, or they nucleate a complex, sometimes referred to as complex 2B, or the necrosome, and we think of just those two proteins together as a kind of necrosome core. That's the subject of our interest here today. A question brought to me by Hao Wu of Harvard Medical School, then Cornell Weill. So let's take a look at the constructs, the full-length domain map of RIPK1 and RIPK3. And by the way, throughout these slides, there's no particular distinction between RIP3 or RIPK3, just terms used in the literature. So both of them have an N-terminal kinase, traditional fold, a lot of homology, has yielded to beautiful structural biology. The group of Shi from Tsinghua University in particular got to necrostatins, very specific compounds that bind to this domain in isolation. And that led to a lot of activity at Glaxo and other places. So beautiful work, particularly on the kinase from RIPK1. And then downstream, a low complexity domain. And that domain contains a signature sequence that I'm going to pay a lot of attention to today. It's a sequence referred to as RIM domain, another acronym for RIP, homotypic interaction motif. You might be surprised to see such a short recognition domain for sequential amino acids, which are similar, not strictly conserved, that mediates important interactions, and RIPK1 and RIPK3 are the classic example. In necroptosis, this is a key interacting region. Mutations here kill the pathway. So we're going to be looking at that interaction, and one of the issues is that in Hao Wu's hands, co-expression in E. coli didn't lead to soluble proteins for the full-length system or various shortened systems around this region, Rather, it led to these nanoscale objects that she visualized in stained EM. Analogous but not identical objects can be seen from eukaryotic cell, various kinds of cells. So here it's a human colon cancer cell line on an immunoprecipitation purified object in the middle there. So apparently only under necroptotic assault pathway being active, these objects appear in cell lines. They also appeared in E. coli. What's the relationship between them? This was investigated in detail in the Wu lab, but also the lab of Francis Chan, the person who actually discovered the role of RIPK3 in the necroptotic pathway. So comparing mutants in terms of signaling in the cell line, in terms of GFP confocal visualization, there's some signature association of RIPK1 and 3, in terms of the in vitro behavior of these E. coli complexes. It becomes clear that we're studying an interaction in vitro that has the hallmark signatures of this complex formation in cells. So one of the questions that came up right away, are these amyloid fibrils? They kind of look like them. So dye binding, fibril, X-ray signatures, high beta sheet content, all of this suggested the possibility that indeed they are amyloids. Let's take a step back and think about how that might be remarkable. They're protective of health, they're human signaling complexes, and they're made of two different proteins. Of course, I think for this audience, I don't need to remind you of what an amyloid is, but here I'll just be precise that here I refer to a structural class. And many of them are in register beta sheets made of strands from neighboring molecules. So it's a very high order oligomer that's always been of interest since its discovery, because it is a hallmark of so many very, very important neurodegenerative diseases. So we're using the typical screens to decide if something is an amyloid. We think it might be, but this became a little more in focus when Ansgar Ziemer came to our lab from Beat Meyer's lab with a lot of experience from his work on the HEAD-S system in that lab, and helped us assign the NMR, solid-state NMR spectrum of these complexes. Now, these are, of course, expressed from E. coli, and we compared full length for both proteins, but subtilized and proteolized to reduce the size and remove some things that don't show up in the spectrum, or smaller constructs that have an area right around the rim domain. These are magic angle spinning CPMAS spectra, and in this case, it's just showing one example. This is an N15 emitic backbone correlated to the C alpha, and you can see enough resolution to start to identify peaks. Other spectra were used to do that backbone walk, not shown here. And what we learned from these early spectra was that there's just a limited region of both proteins that's showing up, but indeed both proteins are in. And moreover, is a very high beta sheet content. And most important to us, the spectra through these different constructs from batch to batch were pretty reproducible. What are we seeing? We're seeing a region that flanks the rim domain, and shown here in these pink boxes. So we only see part of the protein. We only see part of the protein. If we would carry out a J-based or a NEPT sensitized experiment, even though we're sitting in a solid state rotor, we might detect some highly soluble, flexible portions of the protein. If we carry out a cross-polarization and magic angle spinning based experiment, we would selectively detect only those regions that are rigid by a particular definition that on a time scale of the NMR measurements, the angle doesn't change too much, so the dipole coupling still has a substantial time average value. And basically for this and so many other amyloid containing systems, some part is solid enough, ordered enough to yield to solid state NMR CPMAS type analysis above ice temperature, whereas other regions are hydrated and mobile enough that they come and go with temperature a little bit more. So that's actually not that surprising, although it's a little unusual the first time you encounter it. Now, other experiments tuned in to see long-range contacts revealed sequential contacts, buttressing our assignments, long-range contacts within RIPK1 or RIPK3, and really important to us were quaternary contacts between the two kinases. So this, for the first time, made us sure that we're dealing with a molecular species that is built out of two different proteins at a molecular interface. The biology said it should be like that, but these data show us that it is. So those contacts that are in blue, for example, jump from one kinase to the other. Well, we did many different NMR experiments, and most of them were done with somewhat diluted carbons and uniform nitrogen. We can talk more about these pulse sequences, but they come, variously from the Griffin group, some important ideas from Jake Schaefer. Lots of different pulse sequences were employed to get 21 fully unambiguous tertiary restraints and about 400 other kinds of restraints that were either more local or ambiguous. And we used the program from Peter Gunther, Siena, to use these restraints to calculate a structure. One important thing about those calculations is that Miguel Mompian, who came to my lab as a visiting student, working at that time with Douglas Lorentz, MD expert, he took over the structure calculations and quickly realized that we needed no less than 4 copies of each protein in order to represent all the surfaces if we began with a kind of collection of folds that one might entertain either based on the literature or intuitively. And that was an important realization. So we put in eight copies, 4 of each. And in that case, only one fold, only one way of tiling converges without any conflicts. It converges to a structure at the core a little less than 1 angstrom in RMSD, and with no violations. And what drives this structure calculation are a series of contacts that reinforce our suspicion that it is in register parallel. So contacts that connect, and hetero. So contacts that connect RIPK1 to RIPK3, but also some interesting contacts that make a kind of globular interior that go between these signature motif side chains and cause it to fold up and close on itself so that you have this relatively dry and well-ordered interior. So in the end, in this structure, we see a really compact representation of the kind of contacts that have been seen in other amyloids. Ladders of asparagines and glutamines, this intriguing ladder of tyrosine side chains, and something that we think is new or didn't see from other well-defined amyloids, which is a stack of serines and cysteines, an intriguing opportunity there. And we used many approaches to validate this structure. I'm going to have to be, in the interest of time, very brief about this. But first of all, the mutations that attack stabilizing interactions were pursued in cell lines and in vitro in parallel. The cysteine was used as a platform to cross-link. So the prediction is that if we would mutate that serine to cysteine, we could see multimers, and we did. The answer key was used to design a different peptide to crystallize, and finally we got diffraction data that was done by Jixi Li and Hao Wu's group at that time, and now at Fudan, leading a wonderful program in structural biology of immunology. But the real validation in my mind was when we heard very promptly from Margie Sunday at the University of Sydney, who had images like in the top right. She had substituted for the kinase domains some GFPs, and she saw this kind of presentation of a fibril with a dark spot in the middle and all the GFP on the outside, and maybe a slight twist to it. And she saw this for a number of Rim partnering proteins, and our structure was able to explain what she was seeing. That the dark part, we might model in our spine structure determined by NMR, and on the outside, in place of the kinase, co-localized GFPs. Margie has become our partner in pursuing a different question. Many proteins have Rim domains. Many proteins in immune and signaling in our biology. But in one of the, I think, truly wicked things of biology, it is also the case that the very virus that set off this pathway might have a Rim domain at its disposal. For example, murine cytomegalovirus, protein called M45, which turns out to be a ribonuclease reductase, but it has within it a Rim domain, and that Rim domain, it's clearly shown, defeats the necroptotic pathway. And we're now pursuing, Margie, the question of whether it does so by making a different heteroamyloid. So we think of this as a library of heteroamyloids, and we're very excited about taking this analysis to the next level. I'm going to very briefly tell you about a different system. It has in common that it's a key system for cellular signaling and response to assault and controlling cell death pathways. It has in common with the first story that it's a multi-domain protein containing functional domains alongside a low complexity domain. This is the founding member in many cases of the formation of something called a stress granule. Stress granules are thought to provide a mechanism for the bulk of the mRNAs as well as some proteins to be stored or triaged during stress. The formation of cytoplasmic stress granules depends on shuttling a protein called TIA, T-cell intracellular antigen 1, shuttling it from the nucleus to the cytoplasm and aggregating. While many disease-related proteins and their target RNAs shuttle to preformed stress granules, it's believed that the initial stress-induced nucleation depends specifically on TIA and a handful of related similar proteins. And while knockout mice are somewhat viable, they exhibit high embryonic lethality consistent with dysregulation of stress. So it's a very important player in this, and particularly in connection to neurodegenerative diseases like ALS. The RNA recognition motif shown here, RRMs, their structure, dynamics, and the tight, multivalent, and somewhat non-specific binding of RNA have been studied beautifully by solution NMR experiments in Michael Sattler's group. We are most ignorant about the low complexity domain. It's a Q-rich domain, and that's where the locus of mutations that relate to ALS are found. Now, in the literature, most people call it a PRD, prion-related domain. Why? Well, there are many justifications. One is an imperfect but important homology to SUP35. That's a protein familiar to many of you, a yeast eukaryotic release factor, which was shown to propagate as a prion in the 90s by Reed Wickner and became a really important example of transmissibility through his work and that of the late Susan Lindquist and others. It also has a domain structure with a low complexity domain analogous to TIA1. The thing is that truncation mutations of TIA1 lacking this domain, this PRD or low complexity domain, do not induce spontaneous stress granules in cells in the same way that the full length does. And they do not oligomerize in vitro in the same way that the full length does. So more interesting than that, substitution of the PRD from SUP35 can rescue this, can rescue the ability to do this oligomerization. Despite all of this and the biophysical assays of being an amyloid from various studies shown with the citations on this slide, we're going to call it a low complexity domain for the moment. That's what we know for a fact about it. And what we did, and we here is Keith Fritzing, a postdoc in the group who came to my lab from Klaus Schmidt-Rohr and Mei Hong's group with a tremendous education already in solids NMR. We're going to obtain pure full-length TIA1 with good yield from E. coli. That's what Keith did. And then this full-length protein had a solubilization tag, and it oligomerizes reversibly and cooperatively to form a somewhat stable suspension under very mild physiological-like conditions. And so this has the typical lag phase and so on. And watching the transmission, as I'm showing here in the bottom right, the question becomes, at the end of this curve, what are we looking at? This is full length. This is aggregating because of the presence of the so-called PRD, or the low complexity domain. What are we looking at there? Well, a lot of people would like to see an image first. Not us, of course, but full-length TIA forms nanoscale homooligomers. It's pretty heterogeneous looking in the stained EM taken by Keith at the New York Structural Biology Center. But in there, in the midst, you can see these oligomers, maybe 7 or 8 mers. You can see a lot of those, and then you can see other objects that might be the same thing viewed on the side, or we don't know what. The important conclusion is that these are not fibrils. These don't look anything like fibrils. In the solid state NMR of the uniformly enriched material, we saw resolved peaks that we immediately wanted to dig into. And an interesting observation is that if you just count residues, as we often want to do with a new system, it became clear that you could only explain what is popped into the spectrum by saying that RRM2 and RRM3, the two most important domains for function of binding RNA, those are there. Those are there in the spectrum. And by the way, using techniques developed by a number of people, including one of our hosts today, Mei Hong, we can also tell you that those are water accessible. So we got these two domains, they're water accessible, they look folded. Keith's next instinct was to say, can I not assign these by a combination of the usual backbone walk and the nice assignments done in the Sattler group for a very similar protein, but chopped into domains in solution? And it turns out that if you do that, the difference between our shifts and Sattler's are a small fraction of a ppm. It's a match. It's absolutely a match. These are not unfolded. And by the way, if you take the PRD on its own and you decide to study that in NMR, you get a very, very different spectrum. Lots of peaks that we have not been seeing in the full-length system. So what could this be? Our results suggest that the participation of TIA in stress granule formation might not be based on amyloid formation in the C-terminal domain, at least not for the initial reversible steps, because stress granules, due to their function, are reversibly formed. But on the other hand, the data could be compatible with a model like this, where it's a micelle, and there's so much we don't know about it, but we're thinking about the RRM domains on the outside hydrated and ordered based on the NMR and the EM, and then the other domains phase separating to make the thing behave like a micelle, a nice reversible object that can hide things from the cell for a while. So that's an idea. It's our hypothesis. The two systems that I described are similar in their pivotal roles in signaling and sulfate and the domain structure. In both cases, we attempted to step back from the insights that were obtained from divide and conquer. Despite a lot of challenge in systems this large, the NMR spectra offered insights that are like a study in contrasts. For one, only the low complexity domain was visible. For the other, pretty much everything but it was visible. So we look forward to addressing whether these may represent different stages of the biology, how the ligands change or deepen this picture. May said that these talks might, in the best case, inspire us all to get back to work. And here we clearly have numerous questions to look at in the future. So let me end where I started by thanking this remarkable team who set up the series and naming once again some of the key players in this study. Keith Fritzing, here in the lab photo did the gross majority of what I reported on TIA1, starting the project from scratch in my lab. At this moment, Eric Keeler here in the picture and Chen-Ming He are continuing the work on the RIP-K3, but it was started up by Ansgar Zimmer and also Miguel Mompien, now in Madrid, Spanish National Research Council. And the question, which is always the most important thing about a project, was inspired by wonderful conversations with Hao Wu. Thank you so much for this opportunity.
Thanks, Anne. So we have some time for questions, and I see there are already quite a few showing up in the Q&A. So if you haven't found it yet, open up the Q&A at the bottom right of the screen, and for those participating, upvote the questions that you like. So the first I'll read out is from Leif Antonschmidt, and he asks, did you try mutation in order to break certain ladders and potentially abolish formation of the heteroamyloids?
Yes, and I should say those studies were primarily done by Francis Chan and Hao Wu. So I had, if I can skip back to it, I had a list of them, but I didn't take the time to walk through them carefully. This was a very important piece of the overall arc of the project. So in the hydrophobic core, we think of a shape complementarity being part of why the heteroamyloids formed in preference to homoamyloids. And you can ruin that shape complementarity by making some mutations there. We think of the glutamine ladders as being a really important stabilizing element. These amyloids are really hard to take apart. I mean, I know that's a hallmark of amyloids in general. These are a particular case. And they're really dense with these ladders, supporting an idea that's been in the literature for a while that that's an important stabilizing element, even though it's solvent exposed. So we tried to bust up the ladders. We tried to bust up the complementarity in the middle. When we did that, you look in the mammalian cells, for example, the kidney cell line, to see whether the cell-based assays still show necroptotic activity. You can look in the confocal images to see whether RIPK1 and RIPK3 still form puncti together. You can look at the in vitro objects and see whether they've become much less stable. And there was a tremendous correlation, including that the most impaired by functional assays were also the most impaired by biophysical probes of the in vitro. I don't know if that answers the question or if there's a follow-on to that.
Let's see if we can answer a couple more questions as well with the few minutes we have. So we have Shiva Gishukla, if I read that right, who asks, aren't the heteroamyloids by themselves toxic to cells after activation?
They are. So you have a limited time window if you mean mammalian cells, indeed. And you know the usual way of preparing proteins in E. coli, who would know if the cell was sick? We do it late stage and aggressively. So, but yes, I would say indeed. I put the caveat that the reasons in the prokaryote and the eukaryote and the details would be very different. But.
So the next one is, are the kinase activity of both proteins active in the complex that's formed?
That's an important point. So formation of the complex upregulates RIPK3, okay, and so there's various reports about whether RIPK1 can phosphorylate RIPK3 before assembly. But RIPK3 is clearly activated by the assembly of the necrosome, and forming this complex allows it to then phosphorylate the downstream players. So yes, the formation of this assembly is in cells is compatible with function.
Thanks. So I see one more question with a lot of upvotes, and then Christian wanted to answer one, and then maybe we'll have to go to the next speaker. So
Christian's going to answer one. Thank you, Christian.
sorry.
No, ask one, I think. Ask one.
Oh.
Actually, in the meantime, it is two. But the first one is, I mean, the amyloid, is it toxic by itself, or is it whatever bound by something that then downstream kills the cell, or is it a gain of function, loss of function, or whatever? I mean, in other neurodegeneration or so, there.
Thank you.
Always thinks it goes directly to membranes or whatever, and kills them by that. Is that also the case here?
Okay, what is believed and has a lot of support from the cell biology is that this is a signaling complex, and that the hatchet man here is that pseudokinase, MLKL. So the real role of this is to phosphorylate and modulate the oligomerization of that MLKL, which then forms holes in the membrane. So yes, that's helpful to clarify, Christian. Thanks for rephrasing the question.
Okay, so let's do one more. This is probably a quick one, and then we'll go on. So what is the molar ratio of both proteins in the complex? Do they present in 1-to-1?
Yes. Okay, that's a great question. The way that we formed the complex, prepared the protein in E. coli, they were 1-to-1. It was a polycistronic construct. Well, I don't need to go back to that, but it was polycistronic complex to make sure that it was 1-to-1. You might, and it's clear that from the strength of the RIPK1, RIPK3 contacts that there are many interfaces. Do we strictly know that they're checkerboard and odd alternate throughout? No, not at all. And moreover, you can form the homocomplexes. They form, and strongly so. In vivo, they're not expected, in the cell, they're not expected to be equal stoichiometrically. Under some circumstances, you would think you would have a very unequal concentration. So whereas we draw them as strictly alternating, this isn't something we can firmly know based on either the biology or the NMR. I hope that helps with that question.
Okay, so thanks for the wonderful talk. And during the other talk, if you have a minute to read some of the other questions, you can also go in and type the answers there for a few more questions that are in the chat, or maybe stick around to the end. So at that point, I'll turn over the stage for the introduction for Hashim.
I got to stop sharing then. Sorry, Hashim, I'm having an issue controlling this mouse.
Well, while Anne learns to operate her computer, I'll start doing the introduction to save time. My name is Art Palmer. I'm delighted to introduce Hashim Al-Hashimi today. And in particular, in the introduction, I would suggest to students and postdocs listening that this Hashim is a fantastic example of how one should be thinking about managing your career and bringing skills you learn in one area to bear on a new area. So Hashim was a graduate student with Jim Prestigard at Yale, and there did really beautiful work on residual dipole couplings in proteins. I tried very hard to convince him to come to my lab as a postdoc and work on dynamics of proteins, and Hashim thought quite wisely that it would make a lot of sense to postdoc with Dinshaw Patel, learn to handle RNA and other nucleic acids, and bring to bear in nucleic acids the powerful techniques that were being developed for studying dynamics in proteins. In his independent career, he has succeeded spectacularly well at this. You'll hear this today, a wonderful combination of methods development in NMR, applications to tremendously important biological systems, but also bringing into the work a wide range of other biophysical techniques to put together a comprehensive picture of both the biophysics and biological function of the molecules he'll tell us about. I'll embarrass our first speaker by reminding her 25 years ago, we wrote in a review article, development of NMR methods that permit observation of the motions associated with non-canonical structural features is an important objective. And I think you'll hear today that Hashim has just driven right past that sentence on into a very exciting future. So Hashim, we're really delighted you can join us today, and we look forward to your lecture.
All right, thank you so much for that very kind introduction. I do want to correct the record though. What actually happened was Art never gave me an offer. He forgot. Apparently he'll tell you that he forgot about it. So that's why I ended up in Dinchel's lab. No, I'm kidding. I don't want to say that. But thanks, Art, for that really kind introduction. And thanks, Marcus, Christian, and everyone for putting together this wonderful forum. What I'm going to be telling you about today is work by, let me see if I can get my laser pointer here. Yeah, here. So the work that I'll be talking about was mainly the work of Hongwoo Shi and Atul Rangadorai, and as well as a postdoctoral fellow Hala Abu Asi and a new incoming student Rohit Roy. This is a collaboration with David Case, Joe Yesselman, who's a new, who just started his own lab at the University of Nebraska working on RNA folding and design, and also Dan Hirschlag at Stanford, with whom we've had a very long-standing collaboration on determining RNA ensembles and understanding RNA folding. This work is funded by the NIH, and as for full disclosure, some of the technology discussed here was licensed to a company that I co-founded called Namirum. And all of this is an unpublished story, and you can learn more about it in this preprint in bioRxiv. And so the title of the talk is looking at RNA ensembles, and in particular, I want to share with you some of the work we've done recently to determine RNA ensembles at atomic resolution. And I'm not going to spend any time convincing this community that determining RNA ensembles or determining ensembles is important, but instead I'll just remind us for why is this a difficult task. So if we take, for example, a ribosome composed of 1 million atoms or so, and we ask the question, how many parameters do we need to specify the structure of such a molecule? And if you have 1 million atoms and you have to specify XYZ coordinates for each atom, you have something on the order of 10 million parameters total that you need in order to specify the three-dimensional structure. Now let's imagine over here we have a tiny little RNA molecule drawn to a scale with just 1,000 atoms, and you ask the question, how many parameters are needed to specify the range of conformations sampled by an RNA in solution? And if you take the assumption that an RNA samples a new structure every picosecond, and if you assume that the motion keeps going on for a second, then you're going to need to go after Phi times 10 to the power 12. There's 10 to the power 12 structures that need to be specified. And so the amount of parameters that are needed to define an ensemble is on the order of 10 to the power 15. And so the reality is that determining an ensemble of a very tiny molecule is orders of magnitude more challenging than determining the three-dimensional structure of something as large as the ribosome. And I mentioned this to the trainees, don't take the easy path and solve cryo-EM structures of large molecules. Instead, take the harder path and try to solve this more difficult problem of getting at the ensembles, where of course NMR is playing a pivotal role. And so the question is, since we can't possibly measure data on the order of 10 to the power 15, how do we go about thinking about determining ensembles? And the answer, of course, lies in computational modeling. So computational modeling has to be used to fill this data gap. And what I'm showing you here is the generalized scheme that's been used in a wide variety of contexts to get at ensembles of either RNAs or proteins. There are different implementations of this, but this is one that's been particularly popular with RNA. And the idea goes as following. You first take a crystal structure or NMR structure of an RNA of interest, and here's that key step where you run some kind of computational modeling, usually MD simulations, to generate a huge library of conformational states. This is the act of filling the gap by using computational modeling, because we can't ever have enough data to actually build these ensembles from scratch. Now, once you generate this computational ensemble, you'll have some data, typically NMR data, that's ensemble averaged. And normally this initial computational ensemble does a poor job predicting the NMR data because of inaccuracies in the modeling. So the next step involves finding a way to, finding an ensemble from, or selecting conformations from this library, this computational library, that actually does satisfy the NMR data. And there's a variety of NMR experiments that have been used for RNA ensemble determination. These are the techniques that have been used for, experiments that have been used primarily for RNA. And then after that, of course, when you have an optimized ensemble, you see better agreement with the data, and there's usually a step involving cross-validation. Now, of course, this scheme, this idea of using ensemble average data to obtain ensembles has been performed originally on protein, in a lot of studies of proteins, and many in the NMR community have now developed techniques that allow for determination of protein ensembles with near atomic resolution, starting with early work by Blackledge and Bushweiler and Ernst looking at peptides, and then followed by work by Judy Foreman-Kay on globular proteins, and of course all these other individuals who have now contributed immensely to powerful techniques to getting at ensembles of proteins. Now, however, this approach overall has limitations, and particularly for RNA. As you can imagine, if this part is garbage, if you have garbage for a model, you're going to end up with garbage for your ensemble. So garbage in, garbage out. And the question is, how good are these computational models that one is using to obtain ensembles? And so the limitations really are the following. When it comes to nucleic acids, the MD force fields that we use are still underdeveloped. They're also under-tested. And so the accuracy of the ensembles that are obtained by MD are questionable, particularly for RNA. There's also, of course, the problem of sampling. It may be that the conformational ensemble samples timescales on the order of milliseconds. And depending on your protocol, the MD simulation may not be able to sample the entire conformational space. One key issue in nucleic acids also is the lack of data. We don't have as much data typically that we can bring to bear for the ensemble determination approach. The obvious experiments that we don't usually use are chemical shifts and also, for example, PREs. And so because of the limited amount of data, there's limited data to go about cross-validating your ensemble. And so you really don't know how good is your ensemble. Another problem is, of course, that this is a slow approach. It takes years, typically, at least in our hands, to get an ensemble. You have to first solve a structure. That can take several years. You've got to run an MD simulation and, of course, measure all the data. And so fortunately, I have collaborators that are impatient and who think of getting ensembles in years is something that is way too slow. And so in collaboration with Joe Yesselman and Daniel Herschlag, we came up with an alternative approach. We thought, let's bypass this business of structure determination and MD simulations altogether and simply go from the RNA sequence, the one-dimensional RNA sequence, or if you will, the secondary structure, if you have a secondary structure, and directly generate the library using structure prediction. So as you know, there are many programs that have been developed, such as Rosetta, that enable the prediction of protein structures with high accuracy. And such developments have also been taking place in the context of RNA. And of course, the advantages here is that we can literally go from a sequence to a library within a matter of 24 hours. There's, of course, potentially enhanced accuracy because these structure prediction algorithms leverage the PDB to stitch together fragments from experimentally derived structures. So there's the potential for having higher accurate ensembles generated at the beginning. And of course, there's less of an issue when it comes to sampling because there's no timescale limitation with the sampling. You're only really limited by the coverage in the PDB. And so in particular, the approach that we're using is a technique called FAR-FAR, which was developed by Riju Das at Stanford when he was working together with David Baker, and has since further developed the approach. And the key thing that I want to say about this approach is that it assembles together fragments from the PDB and stitches them together to generate various conformations of an RNA target, and then does various rounds of refinement with a scoring function to get at the best lowest energy structure. And in blind predictions, FAR-FAR has really done the very best. And I'm giving you an example here of a blind prediction. You can see that the FAR-FAR prediction here in blue is coming close to within atomic accuracy to the X-ray structure. This is a purely CASP-like competition. And so these techniques have really been developing over the years, and they're constantly improving. So of course, this idea of combining structure prediction with NMR is not new. We all know about CS Rosetta and the work of Ad Bax and David Baker for protein structure determination. Riju Das and Schwalbe and others have developed a corresponding CS Rosetta for solving RNA structures. More recently, Zhang and Dokolian came up with an approach that combines I-fold structure prediction with NMR to get at RNA structures. And of course, perhaps something more closely to what we're doing is Martin Blackledge's work, where he, with flexible Meccano, is able to generate models for IDP ensembles by leveraging the PDB. And so what we really are trying to do here is to generalize this idea of using structure prediction in ensemble determination. And we're going to apply this approach to TAR RNA shown here, which is, if you will, the ubiquitin of RNA molecules, or at least I consider it the ubiquitin of RNA molecules, which is composed of 2 helices and a bulge. And we're going to be particularly interested in the dynamics of this flexible loop, if you will, or the bulge here. And this RNA is involved in HIV transactivation by binding to this big complex. It undergoes the stacking transition when the complex binds and activates transcription of the RNA. I won't be talking at all about the HIV function, but merely demonstrate the approach here on this RNA. So now you might ask the question, how are you going to use methods that have been developed to solve a static single structure and apply them to ensemble determination? And the answer, of course, is whenever you do a structure prediction, you don't predict one structure, but instead you predict a range of structures with various energy scores. So for example, here we took the TAR secondary structure, and literally within a matter of 24 hours and 100 cores, we're able to get a range of conformations shown here with different energy scores. And you can see here at very high energies, we even get stereochemically incorrect structures. But what we're going to do is basically take this 0 energy cutoff and essentially assume this set of conformations here to be the conformational library, which we can obtain again in 24 hours. And so the primary experimental measurements we're going to use are RDCs. And in particular, over the years, my lab has developed an approach whereby we vary the shape of the RNA in order to modulate the alignment of the RNA molecule and measure independent RDC data sets. So you can see here's the wild type TAR. We elongate the lower helix to change so that the alignment is on average parallel to this helix, elongate the upper helix to get this kind of alignment, and so forth. And this allows us then to measure independent data sets using phage as an aligning medium. And we can measure a variety of RDCs targeting, mostly these are the CH and NH RDCs, directly bonded RDCs targeting the sugars and the bases. And in totality, across those 4 samples, we have something like 143 RDCs measured in this RNA molecule. So a few years ago, actually 7 years ago now, we took an MD simulation of TAR on the charm force field on the Anton supercomputer. This was an 8.3 microsecond simulation. And we took this simulation and asked, how good is the MD simulation or the MD generated ensemble right off the bat? How well does that predict the RDCs color-coded for the different helices and the bulge residues? And you can see that there's a correlation, but the RMSD is quite large. 8.6 hertz is much larger than the precision of measurement, which is better than 2 hertz. At this stage, we can do the sample and select to optimize, to select conformers from the MD generated pool to optimize the agreement with the RDCs. And you can see here that at an ensemble size of 20, we can find an ensemble that does better in predicting the RDCs with an RMSD, an overall RMSD going down to 3.6 hertz, and for the various motifs shown over here. So now we wondered, could we replace this ensemble approach with this far structure prediction-based approach? And basically, let's ask, how well does the far generated library predict the RDCs? And you can see right off the bat, the far library, which was generated in 24 hours, in fact, reproduces the RDCs equally well to the MD simulations when we didn't have to solve a structure or run MD. In fact, if you look closely at the RMSD, it's, if anything, a little bit better. The agreement with the far generated library is better than the MD. We can carry out the sample and select approach once again, and we can obtain an even more optimized ensemble using the starting far library. And you can see here, the RMSD is actually better than the MD simulation. In particular, look at the bulge residues. I want you to pay attention here. 3.7 hertz RMSD dropping down to 1.8. And so this was interesting because, of course, we always thought this was, at the time, we thought this was an atomic resolution ensemble. We have done a lot of experiments to validate this ensemble, and yet here we're doing better. And so this was intriguing. You can do cross-validation type analysis where you remove some of the data and only use a subset of the data in the ensemble determination and ask how well do you predict the left out data. And even in this cross-validation approach, whether you leave entire alignment, independent alignment media, or if you remove 20% of the data, even in the cross-validation analysis, you can see that the far library is doing better in terms of the predictions of the RDCs relative to the MD simulations. So we thought maybe this is because the MD simulation, the particular force field that we were using, which is the charm force field, is perhaps not optimized. And of course, there have been advances in force fields over the years. And so what we did is repeated this analysis using a variety of force fields, including the D.E. Shaw most recent force field. And what you can see here, again, here's the bulge RDC RMSD that I mentioned drops significantly for far. You can see that no matter which force field we use, we always are still in this 3 hertz range, suggesting that there's a fundamental limitation in the libraries that we're obtaining using MD, and that far surpasses the accuracy of MD. So of course, now we have an ensemble, a new ensemble called the far NMR ensemble. And the question is, how accurate is it? And is it more accurate than the ensemble generated using MD? And so here we come to the question of cross-validation. And so over the years, we've been collaborating with David Case, who has developed a new approach called AFQM-MM, as many of you know, directed toward predicting chemical shifts on the basis of quantum mechanical calculations of fragments and treating the remaining parts of the atoms, the molecule, using a molecular mechanics-based approach. And so this approach has been developed by David, of course, over many, many years. And recently, we were studying a DNA structure, which we had determined in our lab using RDCs and NOEs. And we took the NOE-derived RDC-driven structure of this DNA duplex and look at the agreement between the measured and predicted chemical shifts for the static structure. And you can see very poor agreement, which is very typical of sort of the agreement that David has been showing over the years about the chemical shift calculations. So we thought, well, let's determine an ensemble for this DNA duplex. So we applied a similar strategy using RDCs to guide ensemble generation of this DNA duplex and asked how well does the chemical shift predictions agree with an ensemble of structures obtained by RDCs of this DNA duplex rather than a single static structure. And you can see that the agreement improved significantly. So this work showed that it's, in fact, it's not David Case's problem. In other words, the calculations are not what the issue is, what's leading to this poor predictions here. The problem is the treatment of the DNA as a static structure rather than an ensemble of conformations. If we use an NMR-derived, RDC-derived ensemble, we do much better with the conformational averaging. And so we thought, can we extend this approach now and attack more complex and more dynamic RNAs such as TAR? And in fact, let's go beyond just the simple chemical shift probes that we used in the DNA study and expand them significantly to include nitrogens and protons and really bring to bear the entire power of chemical shifts to ensemble determination. We have a total of 316 chemical shifts measured for TAR RNA. And so what we did is we took the Anton optimized NMR ensemble and asked the question, how well does it predict the chemical shifts for TAR using the David Case approach? And you can see here the correlation between the C3 prime chemical shift, C5 prime, C4 prime 68 is quite poor. You can see here the C4 prime in particular is pretty bad. So there's very little correlation with this RDC optimized ensemble that we had generated using the MD pool. How does the far optimized ensemble fare out? You can see here, we see dramatic improvement in the C3 prime and C5 prime chemical shifts. And again, for the C4 prime and the C8 chemical shifts. If we ask the question, how does any one conformation from the ensemble, rather than the entire ensemble, picking just one conformation, how well does that predict the chemical shift? And the answer is very poorly. So it's very clear that you need ensemble averaging to accurately predict the chemical shifts. And moreover, this provides additional evidence, independent evidence, because chemical shifts were not used in the ensemble generation, that the ensemble that's being generated using FAR is fundamentally more accurate than the one generated using the MD approach. And if you look, and I'm only showing you a representative of nuclei here, if you look across the board of all the different chemical shift probes that we have, the FAR shown in red always gives you a lower RMSD than the MD and a higher correlation with literally very few exceptions, or no exceptions. Maybe one exception over here. So then the question is, okay, so that this demonstrates that chemical shifts can now be brought to bear in ensemble determination of RNAs. And I would encourage everybody who's either doing solving an RNA structures or determining ensembles of RNAs to use the David Case approach to evaluate the accuracy of their structures and ensembles with this approach. So now the question is, well, what's different about the FAR ensemble versus the Anton ensemble? What is it that's making the data agree better with the FAR ensemble? We first looked at the global conformation of the molecule, the relative orientation of the two helices, which we had studied for many years, and Dan Hirschlag and others had independently verified the accuracy of the Anton ensemble. So as expected, we really see very similar agreement in terms of the ensemble of distribution of the global structure. The two ensembles have very similar interhelical orientational distribution, so this is not what's causing the discrepancy in the bulge RDCs in particular. If you look at the bulge conformation, however, you see differences. You can see here for the FAR ensemble, the different nucleotides in the bulge are more scrambled, whereas you see a more sort of rigid, more ordered conformational states for the Anton ensemble. And so we thought about the sugar chemical shift, the sugar puckers. Given the sugar chemical shifts were particularly bad for Anton, we thought about the sugar puckers. And as you may know, in RNA, we have two kinds of sugar puckers. The C3' endo is the one favored inside helices, sort of the canonical structure. And then if you are more exotic and you're more flexible, you typically can form something like a C2' endo conformation. And so when we look at the ensembles, indeed, we find a very big difference in the sugar puckers. So if you look at the FAR ensemble, it's particularly enriched in this non-canonical C2' and the sugar pucker you see here, whereas the Anton ensemble is very much very deplete in these C2' endopucker conformations. That's true for this bulge nucleotide, for the next, and for the next. So for all bulge nucleotides, you see a diminishment, a much lower population of the C2' endo conformation, driven primarily because in the MD simulations, you rarely sample those C2' endo conformations. Now, is this the reason why the RDC-RMSD is better for FARFAR versus the Anton? So what we did here was we took the FARFAR library and removed all the conformations that had a C2' endosugar pucker in the bulge and asked, how well does the optimized ensemble fare out? And you can see now, once we remove when we only include C3' endo, the agreement goes back to something like we have for the Anton ensemble, indicating that it's the sugar pucker that's driving the better agreement with the RDCs. So now the next question is, well, what would cause the sugar pucker to change? Why is the sugar pucker different in one ensemble versus the other? One thing that we know about in nucleic acids is that in general, when you're intrahelical, when a nucleotide is inside, you assume the canonical C3' endo conformations. When you're intrahelical and stacked, you're C3' endo, whereas when you're extrahelical, you're typically going to assume the non-canonical conformations. And so indeed, when we look at the RNA ensembles, what we find is that the conformation of the FARFAR ensemble features many nucleotides that are extrahelical, shown here in green. So all the green are extrahelical conformations with the C2' endo sugar pucker. And you can see that in fact, the green is always observed for nucleotides that are bulged out, indicating that this sugar pucker repuckering is occurring as a consequence of the bulge nucleotides extruding outside of the helix. Why are the bulge nucleotides extruding out of the helix? They're doing so to enable coaxial stacking of the two helices. The gray here represents coaxial stacking of the two helices. So what's occurring is there's conformational states in the FARFAR ensemble whereby the two helices stack on each other and expel the bulges, and upon expelling the bulge nucleotides, they adopt this C2' endo sugar conformation. And this was very satisfying because in prior work we had shown that in fact TAR is in dynamic equilibrium between a kinked state where the bulge nucleotides are partially flipped in, and a coaxially stacked state where the bulge nucleotides are flipped out. And we interpreted this based on chemical shift perturbations and a crystal structure of TAR, which showed this coaxially stacked conformation with the 3 bulge nucleotides flipped out. Indeed, if we look at our ensemble, we find that we have conformational states that are in near excellent agreement with the crystal structure. So you see here in green are 2 substates from the FARFAR ensemble, and you see the superposition with the crystal structure with this coaxial state with the 3 bulge nucleotides flipped out. What's happening in the Anton ensemble? In the Anton ensemble, you can see there are no conformations where the 2 helices are stacked, and there's a bias towards always maintaining more of the bulge nucleotide flipped inside, likely due to overparameterization, or at least we think due to overparameterization of stacking interactions, which promote intrahelical stacking, and due to underrepresentation of electrostatic interactions, probably that help neutralize the electrostatic repulsion when you form these stacked states. So just to finish in terms of a final test of the accuracy of this ensemble, so if this were true, so our model suggests that we have this cooperative flipping out of bulge nucleotides, shown in red here, for the 3 different nucleotides. You can see all 3 of them flip out when you have a linear conformation, whereas for the sort of intermediate and kinked, you have a more diverse intra and extrahelical states. So we thought if this model was correct, if we could entice the sugar pucker to go towards C3' endo by this 2' methylation, so if we can introduce this 2' methyl, what we would predict is that this would now favor the C3' endo conformation, and as a consequence, favor the kinked state of the RNA. And indeed, when we apply these chemical modifications on either 24 or 23, we see chemical shift perturbations directed towards the kinked states. So, and these chemical shifts are observed throughout the RNA, consistent with a sort of cooperative transition of the molecule rather than independent repuckering of a single bulge residue at a time. Now, I mentioned at the beginning that one of the motivations was that this is fast and the proof is in the pudding. And so here are a variety. So we've applied this method now on a number of different RNAs for which we have determined ensembles. You can see here a variance of TAR with different length bulge nucleotides. And so this is going to allow us now to map the ensemble landscape as we change the sequence context of the RNA. Here's another RNA of interest that Van Herschel's lab has been working on. And this is a very intriguing example where there's a lot of dynamics and base pair melting going on. There are many other systems that we're in the middle of studying. Our goal is to take on all the BMRB all the deposited NMR structures with the deposited RDCs and chemical shift data, and essentially reanalyze them with using this approach and using David Case's approach to test the accuracy of the determined ensembles based on chemical shifts. Okay, so I'll just conclude by running through the different conformational states that I hope I've shown you for TAR, where it coaxially stacks as shown here, with the Thibault's nucleotides flipping out, adopting an extra helical conformation. And all indications are that we are getting closer and closer at defining the dynamics of these RNAs to within atomic accuracy. And so with that, I'll just again acknowledge all the people who did the work, and I hope I stayed on time, and thank you for your attention.
Thank you very much, Hashim. And I forget our organization. Am I going to ask the questions?
If you like, otherwise I can read them out. I also have one if I can use my privilege as a panelist.
Go ahead.
Start out. So, I mean, looking at this FAR library and the workflow that you've done, I mean, it looks like you could use the experimental data behind this FAR library to tune the MD. Are the people that you're collaborating with in MD interested in changing their force fields based on your work?
So with regards to the force fields, I mean, absolutely. I think everybody in the force field community is interested in tests of their force fields and further developments. And actually, David Case, Dan Hirschlag, and I have developed something of a community that's working together toward looking at the force field. And I think the nice thing about the study here is that we're able to pin down particular issues with the force field with regards to the overstacking and intrahelical stacking of the bulge nucleotides. So that's a community effort that's ongoing. With regards to Rosetta, I think maybe you're suggesting, can we bring the data up front or a little earlier in the Rosetta calculation? And we haven't really explored that, but that's a possibility in principle.
And then I'll use my prerogative as your introducer and ask, with regard to simulations of nucleic acids, of course, divalent cations are always an issue. What did you do in the simulations that you talked about, and where do you think those areas need to go?
Yeah, that's a great question. So as David Case will tell you, the biggest challenge, or one of the biggest challenges with nucleic acids, is electrostatics. So a nucleic acid, unlike a protein, has an ion atmosphere associated with it, if you consider divalent metal ions forming both outer sphere and inner sphere contacts, in addition to occupying a cloud around the molecule. In our simulations, we don't have any magnesium or any divalent metals, but that's also true for our NMR experiment, where we didn't have, in this particular case, divalent metals. Instead, the monovalent ions were assumed, and basically you place sodium ions next to each phosphate and run the simulations. So I think that the fact that the coaxial state that I showed you, the actual conformational state that I showed you, was missing in the MD, is the state that's stabilized by metals. So that's the state that is observed in the crystal structure, and I went through this very quickly, but this crystal structure is in fact stabilized by a network of metal ions. And so we think that probably the MD is missing out on this. So these are the metals. There's about 4 metal ions that help stabilize the coaxial state. Now, both monovalent and divalent ions can do this. And so it's possible that in addition to overstacking, there's the challenge in treating the electrostatics is an issue. And Dan Hirschlag and others have mobilized another sector of the community toward looking at calculations of electrostatics. And in fact, David Case has had some good success recently with, for example, calculating the ion binding to quadruplexes and doing experiments of that nature. But I think it's still early. And that's why, for those of you who are interested in pursuing careers, don't think about nucleic acids. There's a lot of interesting problems to think about.
Thanks, Art. Do you want to go ahead with the question and answer, since you're more related to the topic?
Me?
Yeah. Art was asking earlier if you would read out the questions, or me. I mean, go ahead if you'd like.
How do I get to the question? So, okay, I see. Okay. What is the time scale of exchange of the sugar repuckering process? This is from Simon Olson. Is it likely that the initial condition of the MD simulation simply do not undergo this transition? Great question. So we know from relaxation dispersion experiments that all the motions I showed you today, they are R1 row invisible. So these are going to be on the fast time scales, faster than 100 microseconds. We haven't done R1 row Christian-Grissinger mode, that's the next step. But normal mode, 3,000 kilohertz, 3 kilohertz, RF fields, we don't see evidence for R1 row. But I totally agree, Christian, that we should be doing the high power R1 row. So these are what Christian likes to call supra tau C motions that are probably on the nanomicrosecond timescale. Now, with the sugar repuckering, it's a commonly known issue that once you start with a sugar pucker in MD, it really has a hard time sampling alternative sugar puckers. And what we've seen is that in general, either the MD simulation is biased towards C3 prime endo. So even if you start with C2 prime endo, you end up with C3 prime endo. Or alternatively, you start with whatever sugar pucker and the MD is just stuck there and it doesn't reconfigure. Now, we know that sugar repuckering occurs on the nanosecond timescales. So in principle, and we know that from spin relaxation measurements, and so we think that the dynamic should be fast enough, and the MD being microseconds should explore alternative conformational states of the sugar pucker. But I think further work is needed. My PhD advisor asked a question, Jim Prestegard. How long were the MD simulations? Is it possible that it was too short to adequately sample conformations as opposed to problems with the force field? Absolutely, Jim. So the simulations were 8.2 microseconds. And like I said, the motions could be going as far as hundreds of microseconds. And so if you want to sample hundreds of microseconds, you probably need thousands of microseconds or tens of milliseconds to really converge. So that's a very good point. It may not be that the force field, I made the point that I think it's over-parameterization of the stacking, and I say that because of the studies starting from different conformational states. But it could also be that the sampling is not good enough. Now, we did do enhanced MD simulations, and actually those give worse results. So when we do accelerated MD, we actually get worse results with the RDC agreement, and that's because good things break apart when you do accelerated MD. But that's a good point. That's a valid point. Terry Oss, my colleague at Duke, have you ever started the MD calculations from the stacked structure? No, we have not. And that's a good thing to try. Do the FAFR ensembles reproduce the populations of the sugar pucker observed by RD? We didn't observe sugar puckers by RD, but we observed sugar pucker populations using a chemical shift approach. And thanks for the question, because I neglected to mention that in one of the figures that I had, I showed you the sugar puckers. So the answer is yes. Sorry that I can't find the slide now, but the populations of sugar puckers we obtained from the RDC ensemble are in qualitative agreement with the estimated sugar pucker based on analysis of the chemical shifts. And those again show enrichment in the C2 prime endo conformation. Walter Chazen asks, since it is our ultimate goal for understanding function, what are the prospects for integrating emotional properties into the ensemble perspective? That's a great question, and I think many in the field are using, working toward integrating the ensemble perspective to understand function. Dan Hirschlag and I and my trainees wrote a review recently discussing the perspective of using ensembles in understanding RNA folding and function. And I'm sure there are many other reviews written also on this topic for proteins. Should I keep going, or?
Sure.
So, yeah.
Okay. Jeffrey Budenhausen, could proton exchange rates provide evidence in support of sugar pucker dynamics? That's a great point. Proton R1, proton exchange rates. I'm not sure if Jeffrey means amide, immunoproton exchange, or doing R1 row measurements on proton. But either is a definite prospect for looking at as an alternative approach to looking at sugar repuckering, because we do know that there are changes in the proton chemical shifts with sugar repuckering. So if you do high power R1 row on the sugar protons, that might be a powerful way to get at the dynamics of sugar repuckering. The amide exchange, on the other hand, doesn't really tell us much about the sugar repuckering. Andy Bird, do you have methyl probes in the bulge to test dynamics? No, because I'm not Louis K. No. So Louis, as you remember from Louis's last talk, he determined an approach for installing methyl groups in DNA. Actually, if you notice, if Louis is listening, we added a methyl group in our RNA here. So over here, we introduced this one little methyl, which I'm sure Louis is salivating over right now, and these are modifications that are found in RNA naturally. And so, you know, RNA doesn't even have the, I mean, at least thymine has a thymine methyl, but RNA generally has no methyl groups. But this 2'-O methyl is naturally occurring. It's found in many RNAs, and perhaps one can apply a Lewis-like approach where you methylate these RNAs and use them to study the dynamics of large RNAs, and perhaps even get at the sugar puckers in this particular case, although the modification does alter the sugar puckering. Andy likes the idea. Okay, that's good.
So Hashim, you skipped over Katya's question.
I just saw it. I'm going to. Katya Petzel, a former postdoc, how do you interpret the extreme drop of RMSD for bulges for FARFAR 1.8 hertz compared to others? Yeah, so this is a consequence of the fact that, as I showed earlier, it's due to the combination of sugar puckering and the extra helical conformational states. So when we take the FARFAR library and make it look like the Anton library insofar as we remove all the C2' endo sugar puckers so that it's enriched only in C3' endo sugar puckers, when we take such a modified FARFAR library, we end up with an RDC RMSD very similar to the Anton. So it goes back up to 3 hertz. So we can explain the 3 hertz to 1.8 hertz based upon the improvement in the sampling of C2' endo sugar conformations. And that sampling is a consequence of the bases flipping out when the molecule stacks and then becoming enriched in the alternative C2' endo sugar conformation.
Maybe you already answered this. The question from Mark Sanderson, do the J couplings then match the FARFAR results?
We haven't measured J couplings. Well, qualitatively, we've measured the H1', H2' couplings, but we haven't done a more extensive analysis of scalar couplings, and even for that matter, cross-correlated relaxation measurements that Schwalbe and Christian Grissinger have carried out. So I think there's great interest in revisiting sugar repuckering dynamics, and I think there's a lot of different techniques that can be brought to bear to answer that.
And then Chunyu Wang asks about applying these sorts of ensembles to drug discovery.
Yes, so so one of the things that we're interested in doing is using these ensembles and docking in discovering small molecule inhibitors of HIV replication. And we're in the process of now analyzing. So we've shown in previous work that the quality of predictions using ensemble-based docking, one of the interesting things is, when things fail, you always blame the person who's doing the calculation. So when David Case fails to predict chemical shifts, it's David Case's fault. But it's not really David Case's fault. The structure is the problem. It's really an ensemble. I think a similar issue is arising with docking. So, everybody poo-poos docking and says that it's, inaccurate, but, you're typically docking static structures. And what we found is that, of course, there's limitations to docking, but they're nowhere near what you would expect if you really just docked an ensemble of structures. And in one of the studies that we've done, we've shown that the quality of the docking predictions actually improves with the quality of the ensemble as measured by the RDC RMSD metric. And so we're really excited to ask how well does this ensemble now fare out in these docking studies, and we'll see what happens.
And then maybe the last question looks to the future. Shunyu asks, what about single molecule structure determination, such as the free electron laser? So that lets you speculate about it. Maybe the only biophysical technique I don't know you have used yet.
Yeah, so there's been some beautiful work done on using the approach in RNAs, the X-file approach, the laser approaches toward looking at riboswitches, and absolutely, this is a approach that's now gaining momentum in applications to RNA. And so yeah, we'll be interested in also, somebody else should be doing some of that and combining these approaches together to doing these analyses. I think
Lauren, you wanted to comment?
I think only that Christian said he had a question earlier. Is that right?
Yeah, I still have.
I don't think I've ever given a talk without Christian asking a question in my life.
Okay. Now I should prove you wrong, but I will go on anyway. One question is, I hear from my RNA colleagues here that magnesium is required in a lot of cases for functional RNAs, and I think in your cases probably you always remove the magnesium from when measuring because it makes lines broad and stuff like that. But are there any occurrences where maybe the structural ensembles become less? I mean, for example, now that you have the possibility of measuring chemical shifts, you could, of course, compare maybe better the magnesium containing ensemble with the magnesium free or something like that. Any thought direction?
Absolutely, Christian. So I didn't mention it, but one of the ensembles that we have is tar in the presence of magnesium. I didn't show it here, but we have an ensemble, a starting ensemble with one RDC data set for tar in the presence of magnesium. And absolutely, one of the things that we find attractive about this approach is the library from FAR is independent of starting conditions, if you think about it, because it's derived from the PDB. So it will just generate a large library from which you can pull. And in fact, the ensemble that we obtain with the magnesium RDCs is way more coaxially stacked. And it does show more stacked, coaxially stacked. And the equilibrium changes from something like, in the absence of magnesium, the ensemble is 90% kinked and 10% stacked. In the presence of magnesium, it's almost inverted, where it's 90% stacked and 10% kinked. And you're absolutely right that now we can look at the ensemble in the presence of magnesium. We're determining that. And in addition, what's exciting is whether David can take into account the contributions to chemical shifts coming from the magnesium ion. So that's one complication, because magnesium actually interacts with N7s and the phosphates. And by the way, I didn't show you the results here, but the phosphorus chemical shift is not very good. And we think part of the reason for that could be the lack of ions, which are needed to model that interaction. And so I think there's a great prospect of not only getting the ensembles, but like you said, using the chemical shift information to maybe even learn about where are the, down the road, maybe learn about the ensemble of the ions that are surrounding the nucleic acids, and maybe be able to learn something about that as well. But that's, definitely down the road, and absolutely something, by the way, that only NMR, Dan Hirschlag, who's an RNA folder, has really taught me how to appreciate the power of NMR when it comes to pinpointing interactions between metals and nucleic acids. There's very few techniques that can map out with atomic precision where the metal ions are. And so I think that's a whole area that needs further exploration.
I think we should stop there. I'll turn the mic back over to Lauren. And let me end by thanking both Hashim and Anne. Lauren, it's up to you.
Okay, so all I have left then is just announcement for a reminder for the speakers next time. So of course you can go and find the information on the website, but next week's speakers, I believe, are Tua Wang and Rena Rosenzweig, and the following week will be Shimada and Anna Gill. So looking forward to seeing you in those. And if any of the questions haven't been asked, of course, the speakers are accessible through email, and there were some ideas to have other channels as well. So we'll see what forms in the future. Does anyone else have more to say at the end? May or Oscar? Okay, then thanks everyone for joining us again this week, and see you next week.